Hello, I'm Lola.
And I'm Jenny.
Today we want to explain the fundamental principle of DNA replication. We have simplified the process to the basics so that it's easier to understand. You can then deepen your knowledge if you want. First off, we want to revise what we have learned about the DNA in our previous video about DNA. The DNA is a very large molecule. It exists in eukaryotic and prokaryotic cells and in certain viruses, the DNA viruses. The DNA carries the genetic information. The genetic information is encoded in the DNA as the genetic code. All genetic information of the DNA is encoded in its base sequence. The information is the instruction for building RNA molecules. These RNA molecules will, for example, later help to build proteins. This will be the topic of our next videos about transcription and translation. The DNA mostly has the form of a DNA double strand. The abbreviation DNA stands for deoxyribonucleic acid. If you have not watched our video about the DNA yet, you should do that now. In this video about DNA replication, you have to know how DNA is composed.
Why is DNA replication necessary?
The cells in our body are constantly renewed. To do this, a mother cell gets duplicated. It gets cloned, so that there are two identical daughter cells in the end. First, the nucleus is duplicated and then the cell is divided. Like this, one cell splits into two. We have explained these mechanisms in our video about nuclear division or mitosis. At the end of the nuclear division and the following cell division, there are two identical daughter cells that have the same DNA in their nuclei as the mother cell. To achieve this, the DNA of the mother cell has to be duplicated identically first. And this happens during the DNA replication. As we have learned in our video about mitosis, DNA replication happens during S phase of interphase.
And how does DNA replication take place?
DNA replication happens as semiconservative replication. First, the DNA double strand is opened, just like a zipper. Then complementary nucleotides are connected to both strands of the double strand. Like this, one original double strand becomes two double strands that each consist of one original single strand and one new single strand. In the sketch, the new strands are pink. This mechanism is called semiconservative replication.
I understand. Semi-conservative means half-preserving. The two double strands that are created each consist of one preserved original strand and one new strand.
Exactly. Now we want to go into detail and explain the DNA replication step by step. There are several enzymes involved in the DNA replication. These are the most important ones: topoisomerase, helicase, single-strand binding proteins, primase, two different kinds of DNA polymerase, RNase, and ligase. The DNA replication starts at the so-called origin of replication. The opening is also called replication bubble. The first step: at the origin of replication, the helicase has to get rid of the helix shape of the DNA and make it straight. To do this, it rotates the double helix. However, like this, the part of DNA that still has the helix shape, so the part in front of the helicase, gets more and more overwound. The torsional stress gets bigger and bigger with time.
Torsional stress? What's that?
The end of the double strand of DNA is difficult to rotate. In the sketch, this is illustrated by the black bar on the right. The helicase rotates the double strand nonetheless. The double strand gets more and more overwound because the right end cannot rotate.
And how is this problem solved?
The enzyme topoisomerase is needed for this. The topoisomerase breaks one of the DNA strands over and over.
It breaks a strand? Is the double strand not damaged like that?
No, the breaks are reversible. They will be undone again later.
What are these breaks even good for?
The topoisomerase breaks one of the strands after the helicase has straightened the double helix. The sketch shows this. One strand of the double strand stays intact. Here the top one. The other strand is broken. Now it is possible to rotate one part of the double strand, while the other part stays still. The problem of overwinding is solved.
And what happens next?
The next step is also done by the helicase. While it rotates the double strand, it disconnects the two strands at the same time. The double strand is opened just like a zipper. This creates the replication fork. The replication fork is the location of the opening of the double strand. The further the helicase opens the double strand, the further the replication fork moves. In our sketch, the replication fork moves to the right. The following has to be considered. Each replication bubble contains two replication forks. These two forks move in opposite directions.
You said that the helicase splits the double strand of DNA into two single strands. But why do these single strands not connect again immediately? How are they held apart?
Certain proteins are needed to keep the single strands apart. These proteins are called single-strand binding proteins.
Now that the double strand is straightened and split into two strands, new nucleotides can be connected, right?
Before new DNA nucleotides can be attached, a starting segment has to be connected to the DNA strand by the primase. This starting segment is called primer. The primer consists of 6 to 30 nucleotides and is made of RNA.
RNA? What's that?
RNA is quite similar to DNA. However, there are three important differences. First difference, RNA is a single strand. DNA is usually a double strand. Second difference, the nucleotides of RNA are guanine, cytosine, adenine, and uracil. The nucleotides of DNA are guanine, cytosine, adenine, and thymine. So the RNA strand consists of nucleotides, just like DNA, but RNA has the base uracil instead of the DNA base thymine. Uracil is complementary to adenine. Guanine is complementary to cytosine. Third difference, the sugar molecule of RNA is ribose. The sugar molecule of DNA is deoxyribose. This explains the name of RNA. RNA is short for ribonucleic acid. In our sketch, the RNA primer is pictured in a lighter gray. Also note that the primer contains the base uracil instead of thymine. The primers are connected to the DNA strands by the enzyme primase.
But now the DNA nucleotides can be attached.
Yes, the DNA polymerase binds new complementary DNA nucleotides to the two single strands of DNA. The two original single strands are thereby used as a template. But there is an important problem. The DNA polymerase can only read the original mother strand, the template strand, in 3' to 5' direction. For one of the two strands, this is not a problem. The DNA polymerase reads this mother strand from its 3' to its 5' end in the direction in which the replication fork moves. As soon as the helicase has opened the double strand a little further, the DNA polymerase can continue to move without trouble. At this strand, the DNA polymerase does not have a problem. It always runs in 3' to 5' direction. In our sketch, it is the bottom strand. The DNA polymerase builds this new daughter strand, called the leading strand, in 5' to 3' direction without interruption. If you don't know what we are talking about when we say 3' to 5' and 5' to 3' direction, please watch our video about DNA.
Wait a second. You said that the DNA polymerase can only build in 5' to 3' direction. But if the mother strand runs in 3' to 5' direction, then it is not able to work here.
It is able to work here. If I say the DNA polymerase can only build in one direction, I mean the direction of the new daughter strand that is built. This daughter strand is antiparallel to the original mother strand. Since the mother strand runs in 3' 5' direction, the new daughter strand, the leading strand, runs in 5' 3' direction. So the DNA polymerase reads the mother strand in 3' 5' direction and builds the leading strand in 5' 3' direction.
Ah, I understand. This is how you can remember that. The DNA polymerase reads the mother strand in 3', 5' direction, just like we read page 3 of a book first and later page 5.
Exactly. The DNA polymerase can only read in this direction. It can connect new nucleotides without interruption to the leading strand. The construction, the synthesis of the leading strand, happens continuously. However, the DNA polymerase cannot build the other new strand, the lagging strand, this easily. The lagging strand runs in the opposite direction as the leading strand. The leading strand runs in 5' 3' direction. The lagging strand runs in 3' 5' direction.
To work in one continuous piece in the direction of the moving replication fork, the DNA polymerase would have to build the lagging strand in 3' 5' direction.
Yes, but it can't do that.
So what happens now?
This is how the problem is solved. The DNA polymerase builds the lagging strand in small sections. As soon as the double strand has been opened a little further, the primase sets a new primer at the current position of the replication fork. The DNA polymerase then starts there and connects DNA nucleotides to the original mother strand. To do this, it moves away from the replication fork. Like this, it can read in 3' 5' direction and build the daughter strand, the lagging strand, in 5' 3' direction. At the same time, the helicase continues to split the double strand open further. Therefore, the replication fork moves. If the replication fork has again moved a certain number of nucleotides further, the primase again sets a primer at the current position of the replication fork. The DNA polymerase then starts there and connects new nucleotides to the mother strand until it has arrived at the previous primer. Then a new primer is set, and so forth. The DNA polymerase builds the lagging strand in small sections or fragments. These sections are known as Okazaki fragments. In prokaryotes, they are about 1,000 to 2,000 nucleotides long. In eukaryotes, 100 to 200 nucleotides long. The lagging strand is copied discontinuously.
But now there are lots of RNA primers in the new DNA strand. What happens to those?
The RNA primers are later removed by the RNase H. At the same positions, a DNA polymerase connects DNA nucleotides to the strands. This DNA polymerase is a different one than the DNA polymerase we have spoken of before. In the end, the individual fragments have to be connected to each other. The enzyme ligase does this. Now, the DNA replication is complete. 2 DNA double strands have been made from 1 DNA double strand.
In the beginning, you said that each replication bubble has 2 replication forks. Do the mechanisms of DNA replication happen at both replication forks?
Yes, the same procedures happen at both replication forks. But the replication forks move in opposite directions. We have explained the procedure in a very simplified way and left out some details. In reality, the processes all happen at the same time.
This is the end of our video. We hope you liked it.
Here are the links to our last video about the DNA and to our next video about DNA translation.
If you enjoyed watching, please click like and subscribe. If you have any questions or concerns, let us know in the comment section below.
Thanks for watching. See you next time.